dependent in intergeneration mutation instability, and there are many caused by gain of toxic function. The pathological hallmark is intracellular aggregate. And we know mutant huntingtin tends to form beta-pleated sheet and form oligomers, fibrous aggregates. The aggregates are composed of N-terminal huntingtin, proteasome component, autophagal lysosome component, and some of them are transcription factors. And we know beta sheet monomer or soluble oligomers are toxic to neuron, and they cause neuronal loss by the following pathway, including chaperone, proteasome, autophagy impairment, transcriptional dysregulation, mitochondrial dysfunction, increased oxidative stress, excitotoxicity, and neuroinflammation. And neuroinflammation, Dr. Chen, Yizhuang will talk of them in detail later. And before we talk about potential therapeutics, biomarkers are important in terms of being used as an indicator to indicate clinical onset disease progression and to test therapeutic efficacy. Up to now, the current clinical scales are often too insensitive to detect subtle change or subtle progression in mutation carrier, especially those in pre-symptomatic stage. So a useful biomarker is important that could correlate linearly to the disease progression before clinical diagnosis and after 5 stages after phenoconversion. TRACK-HD study has shown striatal volume in MRI imaging can detect progression within 1 year in pre-HD and HD group. So that is the common biomarker now when used in clinical trial to detect the disease progression even in pre-HD group. But we also need biochemical biomarker. And Tabrizi group, they use TR-FLET technology and certain antibody to detect mutant huntingtin in peripheral blood cells of Huntington disease. You can see that mutant huntingtin in monocyte is increased in pre-HD as well as in HD group. However, this mutant huntingtin level cannot discriminate between the early and the late HD group. But mutant huntingtin level in monocyte did correlate with the disease burden score. And the mutant huntingtin level in monocyte also correlated with caudate atrophy. So, and then wild group, they use single molecule count immunoassay to detect CSF mutant huntingtin. Here you can see CSF mutant huntingtin is increased in pre-manifest HD, also in manifest HD group. And the CSF mutant huntingtin level correlated with disease burden score, also correlated with UHDRS motor score. So it seems that CSF mutant huntingtin is a good marker. And as we know that NFL has been shown to be a good marker for several neurodegenerative diseases, including Huntington disease. And this study shows that CSF plasma NFL is correlated to CSF NL level. Plasma NFL level is increased in pre-manifest HD, also in manifest HD, And the level as correlated to the clinical severity of HD also correlate to the striatal atrophy. And this this NF level in plasma, the basal level can predict progression of pre-HD within 3 years. And this level also correlate to longitudinal change in cognitive motor functional decline and also correlate to brain atrophy. So up to now, biofluid, mutant huntingtin, and NFL, along with caudate atrophy, are among the earliest detectable change in Huntington disease. So we have some useful biomarkers in clinical trial, and I will talk about later how they use in clinical trial. Up to now, there is no disease, there's no therapeutic to prevent progression in Huntington disease, but we do have some symptomatic treatment. As you can see here, tetrabenazine is a VMAT2 inhibitor that has been approved by FDA to reduce chorea. And we can also use some anti-dopaminergic drug to treat chorea and psychiatric symptoms. Tetrabenazine is useful in reducing chorea burden, also reducing chorea severity. However, it did have some side effects, such as increased risk of depression and suicide ideation. Recently, FDA have approved another drug called deutetrabenazine. Deutetrabenazine has the same structure as tetrabenazine, but the hydrogen is replaced by isotope hydrogen. Deutetrabenazine has a longer half-life and less side effect than tetrabenazine. And we now going to talk about some potential therapeutics that can prevent disease progression. So these figures show you the pathogenic pathway involved in Huntington disease. So the first, there is transcriptional dysregulation and inflammation, excitotoxicity, misfolding protein, abnormal protein load, and lack of BDNF trophic factor, and mitochondrial dysfunction, oxidative stress, also autophagal lysosome impairment. So we can target this pathway to develop a new strategy for Huntington disease. Here you can see mutant huntingtin impair phosphorylation of CREB, impair acetyltransferase activity of CBP. And mutant huntingtin is dissociated with REST. REST then bind to NRSE to inhibit BDNF transcription. So HDAC inhibitor can enhance histone acetylation. BDEI, phosphodiesterase inhibitor, can enhance CREB phosphorylation. And clinically, Pfizer just launched phase 2 for PDE10A inhibitor, but for HDAC inhibitor, phenylbutyrate just failed. The phase 2 of the phenylbutyrate results are not published. And the cysteamine is a transglutaminase inhibitor, also BDNF release enhancer, but it fails in phase 2 clinical study. And we know that mutant huntingtin can interfere with CREB, TF4 mediated transcription. So leading to impaired PGC1 alpha and PPA gamma expression that lead to decreased mitochondrial biogenesis and mitochondrial dysfunction. So agents enhance PGC1 alpha and PPA gamma expression or activity, or such as cyclosporine decrease membrane permeability of mitochondria, can enhance mitochondrial function. And this study has been shown to be effective in animal study, but clinical trial creatine coenzyme Q failed in phase 2. And resveratrol just finished their phase 2 study. And triherpetanium is a tri-TG that may reverse metabolic deficit in PD by supplying substrate to Krebs cycle. And the clinical study is just, phase 2 is just ongoing. And there are several or many antioxidants has been used to animal study to show their therapeutic effect. And there are several listed here. But dimebone is a strong, very strong antioxidant. Dimebone failed in phase 3. And also diol, also there has been completed in phase 1, but the result is not clear. And this study is using pridopidine. Pridopidine is a dopaminergic stabilizer that can be considered to be a dual-acting agent. They also display dopaminergic transmission antagonism, but also strengthen corticoglutamate transmission. That also binds to sigma-1R. That is a chaperone strongly associated with ER and mitochondria. But pridopidine in phase 3 also failed. It did not change any primary endpoint or any functional asset. However, 90 milligram per day, that pridopidine did reduce tertiary endpoint. That is motor scale, total motor scale. But pridopidine need further clinical trial to confirm its efficacy. And anti-inflammation agents have been shown to be effective in animal study. Here list a few agents. Laquinimod has complete phase 2 trial, but it did not meet a primary endpoint. But it did reduce whole brain atrophy in early stage of HD. And anti-semaphorin 4D just completed, just in the phase 2 trial. And you can see monocycline failed in phase 2. Cannabidiol also completed its phase 2 study, but the results are not known. Chaperone can help to refold the misfolded protein. So chaperone maybe have some therapeutic potential, but there are none of them going to clinical trial. When the misfolded protein accumulated, ubiquitin proteasome pathway is activated. E1 activate ubiquitin, E2 conjugate ubiquitin, and E3 ligase bind to substrate for polyubiquitination, and then send to proteasome for degradation. When the abnormal protein load is too much, autophagosomal pathway will precipitate. Then the ubiquitinated protein bind to P62. P62 is autophagy adapter bind to LC32. Pro-LC3 is activated by ATG4 and ATG7 to form LC32. LC32 is critical in autophagosomal formation. And ATOC3 is representing autophago lysosome activity. So that is a marker for autophago lysosome. Trehalose has been shown to be effective in reducing aggregation, reducing brain atrophy, and improve rotarod performance in R62 mice. Subsequently, several molecules that enhance autophagy has been shown in a cell study, but none of them, including trehalose, has been gone to clinical trial. And recently, we have some strategy to selectively degrade a protein of interest. We call it a pathogenic protein. The strategy is to drug undruggable target. There are many two approaches. One is proteasome tag chimera, one is autophagosome tailoring compound. Here show you 4 systems utilizing indicated E3 ligase, that is E3 ligase. The protein bind to individual E3 ligase and their protein of interest, then indicate and induce polyubiquitination of the POI, then bring the POI to proteasome for degradation. Tomo CG in Tokyo has a synthetic compound that has been used as a linker to link CIAP1 and polyubiquin and mutant huntingtin, and bring the mutant huntingtin into proteasome for degradation. They have shown this molecule decrease mutant huntingtin in HD fibroblast. Another way, attack autophago tethering compound, attack link to LC3 and the mutant huntingtin, and tether the complex into autophagosome for degradation. And Li from China has shown has identified several molecules that can bind to LC3 and specifically mutant huntingtin, not wild type. Then this complex went to going to autophagosome for degradation. They also show that this attack can improve phenotype in cells, in fly, in mouse model of Huntington disease. Let's talk about there are molecular therapies targeting huntingtin production. I just talked about PROTACs. So we can use CRISPR-Cas9 zinc finger with talents to cut CAG on genomic level. We can use ASO to hybridize to pre-messenger RNA to induce RNase degradation of pre-messenger RNA. We can also use microRNA or small interference RNA to bind to messenger RNA to induce risk-mediated degradation of messenger RNA. So now we have several clinical trials ongoing. First one is Ionis Pharmaceuticals. Their ASO has just completed phase 1/2A study. And their ASO is non-allele specific. WaveLife company just in the phase 1B and 2A clinical trial. Their ASO is SMP specific. This one is also allele specific. They target on CAG repeat expansion allele. ASO must be administered by intrathecal injection. their action is short, need to be repeatedly administration. Talk about RNAi, microRNA or siRNA must be carried by AAV and delivered through intracranial injection. However, their action is long-lasting and irreversible, and they only need to be injected once. The clinical trial for RNAi, they just in the preclinical study. Some of them are just begun their phase 1 study. And there are also small molecule splicing modulator to modulate splicing site, but they are only in the clinical, preclinical study. This one is ASO from Ionis Pharmaceutical. They call the ASO called HTTRX. And HTTRX injection, intrathecal administration can reduce CSF mutant huntingtin level in a dose-dependent manner. And they also show reduction of mutant huntingtin level correlate to improved UHDRS score. Since the phase 1 and 2A has shown positive result, this SO treatment is under phase 2B and 3 studies. And the last one, I talk about transcription level. SPT4H is required for expression of expanding CAG-containing gene, but not for wild-type gene. So if we can delete or knock down this SUPT4H gene, maybe as potential for the treatment of Huntington disease. Professor Zheng from Yangming University has shown knockdown of SUPT4H decreased mutant huntingtin level, but not wild-type huntingtin level. And they also show the depletion of SUPT4H improved phenotype and survival of R62HD mice. Therefore, inhibition of SUPT4H reducing mutant huntingtin aggregation and toxicity, while not altering overall cellular messenger RNA is a potential therapeutic strategy for Huntington disease, but they not yet develop any molecule for clinical trial. So take-home message is that Huntington disease is a gain of toxic function involving several pathogenic pathways. Targeting on those pathogenic pathways, we may have developed some potential therapeutic strategy. And useful biomarkers are important. Now there are about 4 biomarkers that can be used in clinical trial to test the therapeutic efficacy. No treatment to prevent disease progression now, but we do have some symptomatic drugs, such as tetrabenazine, deutetrabenazine. PDEI, anti-inflammatory mitochondrial function enhancing agent, are under clinical trial. Antisense oligonucleotide is one of the most potential therapeutic strategy. Success in phase 1, 2A, and is under phase 2B and 3 clinical trial. Other HTT lowering strategies, such as PROTEC, ATEC, and gene editing therapy are now potentially beneficial and under development. Thank you for your attention.
Talk. Any question from the audience? Okay. May I have the first one? Oh, sorry.
Right. Thank you. Really a very comprehensive talk. As a basic scientist, it's really pretty sad to find that many studies that have been very effective in the animal but not effective in human. So do you have any suggestions or advice for us to, when we are interested in developing drug, and what are the criteria that we must have to see in animal study before we go on to human study?
That is a tough question. I think the many differences. There is difference between mice and human. And I think most of drug didn't really go into brain. The BBB permeability is mainly a factor. And such as talk about tree hollows, because we have strong many tree hollows to digest tree hollows. So I think that is why tree hollows didn't go to clinical trial, because once they go into the gut or go into the blood, we have tree hollows to digest. So we may need to consider several. There is a huge difference between animal and human. But I think penetrate BBB and confirm there's enough dosage of the tested or the tested drug go into the brain is very important.
Okay, may I have the second question? There are several ongoing clinical trials, and in your opinion, which one will be or can be expected to have a promising result?
Up to now, the most promising is the antisense oligonucleotides, because they show reducing mutant huntingtin, and the reducing mutant huntingtin correlates to improved clinical rating scale. But that is only a few patients, so they need to see a bit longer or a larger samples.
Please.
Thank you for the comprehensive review. I have a clinical question. Do you think among the several choice, even in clinical trial or not yet, the patient's CAG repeat number or their age of onset, where this will have impact on the response to this treatment? Or we need to choose different therapy depending on the patient's CAG repeat number or age of onset?
I think a larger CAG repeat is associated with more severe clinical features and more progressive disease course. So we talk about treatment. We like to see, we like to apply the drug as early as possible, and we can divide the patient severity into mild or into severe group that based on CAG repeat length. I think that is a good idea.
Any clinical trial result where the CAG repeat will influence on the patient's treatment response?
Now they most choose not so severe. They tend to select less severe patient go into clinical trial first.
Thank you, Dr. Chen. I have a short question about the cognition in Huntington disease. Because I have a patient, he had a severe motor disability, but almost normal cognition. So I have a question about how to predict those patients will have cognitive decline, if there are any biomarkers and any trial about the treatment, about the cognition in Huntington disease.
Regarding cognitive treatment, there is no FDA-approved drug, but you still can try agent or drug treat for Alzheimer's disease. I think the level of acetylcholine is also lowered in these Huntington disease when they have dementia. So you may try the agent such ACE inhibitor in those patients, but that is not approved by FDA. Also, any treatment for dementia, exercise, or biomarker, we can say NFL. NFL is a biomarker for dementia. So the NFL also, I have shown the level also correlate to language ability and cognitive function. So maybe NFL is a marker for dementia.
On behalf of the Taiwan Movement Disorder Society, I will present a paper of appreciation to Professor Chen to thank his contribution to this Congress.
Due to time limit, it's now my turn. Hello everyone, my name is Chen Pei Hao from Makai Memorial Hospital. It's my honor to introduce the next speaker, Dr. Chen Yizhuang. Dr. Chen Yizhuang received her PhD in molecular and cellular biology and postdoctoral training in signal transduction from University of Massachusetts and Harvard Medical School. She is currently a distinguished research fellow in the Institute of Biomedical Science at Academia Sinica, and her laboratory focuses on development of novel therapeutic treatment for neurodegenerative disease, including Huntington disease. In addition, Dr. Chen has published over 120 SCI peer-reviewed papers. Today he will present a topic that I ever don't know what he's talking about. Microglia galectin-3 as an innate immune response regulator in the brain of Huntington disease. It seems as a neuroinflammatory marker. And let's welcome Dr. Chen Yizhuang.
Good afternoon. I would like to start by thanking the organization committee, and also it is a great pleasure and honor to speak after Chongmei. We have been collaborating for a long time. So also, during Chongmei's talk, you guys already have a comprehensive presentations on Huntington disease and also how this disease is treated. Could I have my slide? Okay, our chairperson mentioned that galectin-3 is is not known at all. And I think then this is a good idea that we talk about it, because this is something that we still have room to work on. And Taiwan, we have many scientists working on galectin. Okay, let me briefly introduce Huntington disease. Of course, that we don't have to introduce it in this talk. But I do want to mention that except in neuron, actually mutant huntingtin is expressed in 22 different tissues. So I remember, as Chongmei just told us, that the most promising clinical trial at this time is antisense oligo. And about 2.5 years ago in United States, in Boston, there was a talk discussing, because now they are preparing go into phase 2 or phase 3 study. At that time, that the scientists involved show many good data about it. And the clinical trial already started the phase 3 last year in January. And today they already, although there are a pandemic and everything, they already collect 800. Is that okay? Right, okay. They already collect 800 patients are not doing antisense oligo. But think about it. There are two things that people still have to consider. One, as Chongmei had mentioned, for the antisense oligo, it goes into intrathecal. So it's a very heavy burden for health worker. So the patient has to go into hospital every 2 to 4 months. So that's one thing. It's a little bit difficult. And because of that, the cost would be high, right? And you have to do it throughout your life. That's one thing. Another thing, as we mentioned, mutant huntingtin actually expressed not just in neuron, also in microglia, and also in liver and other parts. So if we only do downregulation in the brain, is that going to solve all the problem? So those are the two things that are being discussed in the society. Now, because of that, we are thinking maybe we should think about whether there are new things that we should look into. And that's also related to the question that I just asked as a basic scientist. How do we know what we're doing really would be useful for clinical study? Now, Huntington disease, of course, is very important because many big pharma company, they actually look into Huntington disease, not really because this disease has a huge market. No, we all know this is a rare disease, but because it actually would provide an opportunity for drug to test first in Huntington disease. And as Chongmei mentioned, the important thing is the drug has to go into the brain. In that case, if a drug working on a common disease for Huntington disease, later it might be able to be applied to Alzheimer's disease, for example. Because the problem that people now are facing are very similar. Now I want to introduce galactine. This is a very interesting family of protein that it basically is a family of protein that bind carbohydrate. All right, this sounds a little bit unfamiliar. However, think about it. On the cell surface, protein, lipid, they all have carbohydrate, different carbohydrate. And when microglia, neuroinflammation, need to recognize the degenerating neuron, sometimes they base on surface carbohydrates. Okay, so this is a family of very interesting function. All this you have seen, those are domains that bind to carbohydrate. They can be monomer or dimer with a linker that indicate they probably could link two molecules together. And galactine-3 is our focus today. What it has is that it has a carbohydrate binding site, but also with another domain. Now, the reason that we think this is important is based on this mechanism, we might be able to think of something to treat disease. That's the goal. And also another thing is very interesting is that galactine-3, although it exists in many parts of the brain, its level is really low under normal condition unless that the microglia is upregulated. And therefore, if you are using an inhibitor to inhibit its overactivation, it could be safer than the expression is higher. All right, another thing is also interesting. Look at this, ALS, Alzheimer's, epilepsy, this is another rare disease, even TBI. When there's a trauma in the brain, galactine-3 level is increased in the brain, sometimes even in the blood. So this makes it an interesting molecule that we really want to look into. How about Huntington disease? So these are my two good friends. We've been collaborating more than 10 years, Dr. Chen Chongmei and Dr. Song Bingwen. So our first question is, as we all know, when the patient comes to your clinics, they already know, or probably will know soon, whether they are carrier or not. They would do gene tests, they would know that. One thing they don't know is when the disease will be onset. So we're thinking, can we actually look into their brain first? For example, that if we measure the galectin-3 level and then compare, this is control subjects, carrier for these, and this is really onset, we saw significant difference, statistically difference. And then if we follow, for example, here we look at MMSE, we know that this is not really the cognitive function that we are looking at, but it also correlates with the galectin-3 levels. If we look at the disease burdens, the motor function, the independency is all correlate. So to us, this is a very important information that in addition to the mutation, maybe the blood galectin-3 also is an important marker. But in order to make a treatment, we need to know where it happens and how it happens. So the next thing we do is to look at the brain, human brain, normal subjects, and the Huntington's brain, and the galectin-3 level in the brain, and we found there's an increase. So the upregulation not only occurred in the blood, also occurred in the blood, also in the brain. So that makes it something that we can work on. So the function, and also that related to what does that mean? So in order to study that, the first thing we do is to find a good animal model. In this case, is a transgenic mouse model of Huntington disease. And very similar to human brain, you will find the galectin-3 level is increased throughout the disease progression. So this is pretty good. It can be a good model. And if we look at the Western, we also found the increase in the Huntington's mouse. Where is this molecule? So we do a very simple staining, and we found this is a galectin-3 level, the green one, and it co-localized and only co-localized with about 1 positive, which is a marker for microglia. So the intensity in microglia significantly increased in Huntington's mouse. Is that a good thing? Is it activated, or maybe it's just a marker? For that purpose, we also stain this one is for the primary microglia. We isolated primary microglia from Huntington's mouse and look at whether they also express galectin-3. If you compare wild type versus HD, you'll find the galectin-3 level is significantly increased. That indicates that really it doesn't require other cell type. Simply microglia would give you high galectin-3 levels. Okay. Now, neuroinflammation, if we threw out the keyword, people would normally think about NF-kappa B pathway. So the first experiment we did is to see whether the upregulations of galectin-3 is related to NF-kappa B. So we use an inhibitor in here. Now, all these studies were quantitatively analyzed. So let's look at the image that in the brains, in the microglia of wild-type animal, there is almost no galectin-3. And the level is extremely high in Huntington's microglia and is significantly suppressed by NF-kappa B inhibitor, indicating that, yes, this is NF-kappa B dependent pathway. Is that related to neuroinflammation? For one thing, let's look at the TNF-alpha. We all know is a marker for neuroinflammation. Then we could suppress it by suppressing this NF-kappa B pathway. It also increases a pro-inflammatory cytokine, which is IL-10, shown in here. So this is related to neuroinflammation. But then the next question is, then what does galectin do? Does it do anything? Is it important? So that comes my collaborator Professor Fu Dongliu. He has been doing galectin-3 for many years in the world. And because of that, if you are interested in doing similar type of galectin study in your disease, Dr. Liu is very, very helpful in collaborating. Now, so if we look at this, here again, the green signal indicate microglia galectin-3. Their lab provide a very effective tool to suppress galectin-3. Now, when this is suppressed, you will find the NF-kappa B pathway is also suppressed. So naturally, this can be a new route to look into neural inflammation. So again, that we look into TNF-alpha, and we found if we suppress galectin-3, you would find the neuroinflammation TNF-alpha level is reduced. Another very important thing to look into is whether it's intracellular or extracellular. Again, you probably think this is too much detail, but in order to have a good design for therapeutic treatment, this can be important. So we look into whether it's intracellular or extracellular, and thanks to my collaborator, Dr. Lin Junhong, he synthesized an inhibitor that is cell permeable to galectin-3. So let's look at this inflammatory mediator TNF-alpha. You'll find in the Huntington's microglia, if you treat it with TD139, it would suppress it. So this site is probably intracellular, because if we use another treatment, lactose, a very simple compound that cannot enter the cell, but could block the function of galectin-3, you find there's no difference at all. So what it tells us is that first, we see a feedback we see a regulation of galectin-3 to control the NF-kappa B, and its function is intracellularly. Now, another interesting thing is that if it's intracellularly, where does it happen? So we did a whole panel of staining, and we found here, again, the galectin-3 is the red signal, and only in lysosome, LAMP1 is a marker that we found it co-localized. We send lots of marker, for example, mitochondria, autophagal zone, etc. There's no co-localization except in the lysosome marker. And if we look at that, this is another image just to showing the same thing, that you would find galectin-3 here, LAMP1 here, they totally co-localize. And if again that you're looking at 3D construction, it's very clear that lysosome just wrap around the galectin-3. When we're trying to submit this for publication, our reviewer actually were not happy about it. They asked that we should also do EM. So this is just one example that we are using EM to look at two things, LAMP2 for lysosome, galectin-3 for their colocalization. So in wild type, you will see the, here is the lysosome, and there aren't too many galectin-3. Only here, and it's outside of lysosome. But if you look at the Huntington's microglia, you find lots of very small galectin-3 in the lysosome, and showing that they are really true, reflecting what we have seen in the immunofluorescence study. So we don't understand why this happens, and there's another very important study report by Dr. Huang Jierong in National Yangming University. He report something initially I don't understand, that he said that liquid-liquid phase separations are very important for galectin-3. What that means is that the protein will actually form a assemblage that in the cell. And those can be quite important. And for galectin-3, he believes is in the C-terminus, the N-terminus. So the question is that if galectin-3 is happens in the lysosome, what does that affect the function? Does that affect the function? So lysosome, that we know the function, this is almost like a garbage truck for our cell. Most of the bad material that it eat up will be digested in lysosome. So if lysosome is damaged, it cannot degrade the thing that it needs to degrade. And also, if it's broken, the enzyme inside the lysosome will be leaked out into the cell. Now, today, that if you check the medline, you're using lysosome, and let's say Alzheimer's disease, many degenerative disease, you'll find the functions of lysosome is important for most of the degenerative disease. Now, so the first question we have is, if Huntington microglia has lots of lysosome, are they impaired? So we are using an assay, a dye assay, to highlight the damaged lysosome. And you will see just by imaging that the Huntington's microglia, they have upregulation of the red signal indicating their lysosome is impaired. And this can be blocked by a galectin-3 inhibitor. So again, we have a lot of analysis and indicating that, for example, that lysosome's mass is increased in HD's microglia, indicating they have swell, larger lysosome. And what does that mean? That indicating that they are probably broken but cannot be digested by autophagosome. So that get into the cell even become more inflamed. Okay, we can talk about that more if you're interested. But the most important question is, of course, whether this is a therapeutic target. So what we did is that, again, using a knockdown approach, we first make sure that we can knock down galectin-3, and by doing that, we knock down the microglia activation using the activated marker. And then we could look at, for example, inflammatory marker IL-1 beta. We found the inflammation is reduced in here compared to this. And of course, we could also look into, most importantly, it increased slightly the survival and also improve the motor function in mouse model. Okay, so this is basically what our story is. We show that mutant huntingtin is being well known, that its presence would enhance copper B signal, and it would trigger the galectin-3 level to increase, and the upregulation of galectin-3 actually facilitate the inflammasome-related neuroinflammation, produce more cytokine, and cause neurodegeneration. So it's a new therapeutic target that we can probably consider, because if we remove it, it actually rescue Huntington disease. Now, another thing is that we believe it's probably also important for other diseases. Now, by saying that, I will show you one piece of data that we all know. Alzheimer's disease has two markers. One is the tauopathy, one is A-beta amyloid. So there are no good animal model for this, but if you look into their separate model, for example, APPPS1, I think some of our audience may have used it, and also tau overexpressed mouse model. These two mouse, they all show cognitive impairments. So they are reasonable animal model. Now, if you look at the green signal, which is galectin-3, for example, here, this is a tau animal model, or the gray signal is a tau hyperphosphorylation, and galectin-3 really, the positive cell are here. So they are not neuron. So what are they? they are co-localized with ABA1 signal, indicating they are a neuron. And very similarly, we saw lots of upregulation of galectin-3 in the APP-PS1 mouse model. And if you look at the ABA1 signal, you found really this galectin-3 signal is from microglia. And therefore, in the future, if we have a good anti-galectin-3 inhibitor that could pass the BBB, enter the brain, it could probably be applied to many disease or trauma that are related to galectin neuroinflammation. All right, so with that, I would like to thank all my collaborators, and also most of the work were done by Dr. Jianjing Xiao, the student that just graduated. So thank you so much, and we would appreciate comment or suggestion.
That may be involved in galectin-3 activation.
Right, that's really one of the key data we've been working on, and the inflammasome 3 that we have looked at the marker expression and also the inhibitor. We also believe the galectin-3 up, the promoter part would have probably NF-kappa B elements. So at this time, that seems the most likely pathway. But one thing that I didn't have time to talk about is the Panta formation, the LPS things. Those are probably because the N-terminus would allow them to form a protein aggregation. Because of that, they are difficult to be removed. So that is also important for the galectin-3 pathogenesis, I think.
Thank you.
Any other questions or comments? Okay, over there.
Thank you, Professor Chen, for a wonderful talk. I just wondering, because the Dr. Chen has first mentioned about, also we find the plasma galectin-3 is also elevated, and as well as in the CSF, data from animal study, right? I wonder, does the peripheral galectin-3 de novo occur in the periphery, or it come from the central? Because I think if we systemic injection of galectin-3 inhibitor, does it also help for in other systemic symptoms, not only in our CNS?
Right, thank you, great questions. Actually, that was one of the questions our reviewer asked. They want to know the source of peripheral galectin-3, and they are because in Huntington disease, for example, people already know macrophage, B cell, T cell, they are all upregulated, the cytokine inflammation. So at the end, that we believe is it could probably from peripheral as well. So like you said, if we systemically give the animal or patient later, hopefully, that for galectin-3 inhibitor, we should be able to treat both peripheral and central. And today, they are already galectin-3 inhibitor on phase 3, but for some lung problems. they cannot enter the brain. So we are also doing a project trying to select those galectin-3 inhibitors that could enter the brain. But I think you're absolutely right. We are now looking for a systemic treatment for this disease. Yeah, thank you.
Okay, any questions? Okay, on behalf of Taiwan Movement Disorder Society, we'll like to present Dr. Chen a gift of certificate of application.
Thank you. I hope you now would remember galectin-3.
Okay, let's move to the last session. The speaker will be Dr. Liu Youchen from Taipei 荣民总医院. Dr. Liu graduated from National Taiwan University Hospital, and she completed her neurology residence at Taipei Veterans General Hospital. After winning the studying abroad scholarship supported by Taiwan Ministry of Education, she got her PhD degree at Institute of Neurology, University College London. Her study focused on neurogenetics and functional imaging. She is now a neurology consultant at the Department of Neurology, Neurological Institute, Taipei Veterans General Hospital, and associate professor at National Yangming University School of Medicine. She used different tools of neurogenetics and functional imaging to explore the pathogenesis of several paroxysmal disorders, such as paroxysmal dyskinesia and epilepsy. And now she's given us a talk on motor circuit dysfunction in paroxysmal kinesiogenic dyskinesia. Dr. Liu, please.
Thank you for the chair's introduction, and also thank you for the committee to give me the opportunity to share some of my studies. I'm Yosen Liu from the Taipei Veterans General Hospital. Today I will talk about the motor circuit dysfunction in paroxysmal kinesiogenic dyskinesia. First, just give a very quick clinical overview about the disease. Paroxysmal kinesiogenic dyskinesia, we call it PKD, also termed episodic movement disorder. It's characterized by involuntary movements triggered by change of postures, like initiation of standing, walking, or running. The attack actually may consist of a variety kind of involuntary movement, including, including the dystonia, chorea, or mixed. And the involvement may be unilateral, bilateral, or alternating. The onset is very early, usually in the childhood or early adolescent. The duration actually is very short, within seconds to 1 minute, but the frequency may up to more than 100 per day. But the patients always preserve their consciousness. So conventionally, PKD is regarded as a kind of movement disorder, which involves substantia nigra and dorsal vagus. Please start the video. This is just the patients. We recorded the attack during admission, and you can see we asked him to do the jumping repeatedly. I think there's some lagging about the video information. We're trying to force him to repeat the jumping continuously, and there's some problem. Okay. maybe we'll come later. We try to ask him to repeat the jumping with his left leg, but he has difficulty to do the repeated jumping. So finally, he fell down and got injured. So we maybe jump to the next slide. Yeah, sorry. Sorry for the problem. But you can see that actually the PKD, although it's a kind of paroxysmal disorder, actually he will cause the patient to get injured. Okay. So we know that PKD is viewed as a basal movement disorder, but very interesting, it has been a long time we noted that there is some clinical overlap between PKD and epilepsy. Many patients have infantile epilepsy, and their families sometimes it's common to have seizure disorder. And very importantly, the treatment for PKD is give them some anti-epileptic drug, particularly some sodium channel blocker. So it's also hypothesized for a long time that the clinical overlap may come from the genetic overlap. We have already known that in some region on the chromosome 16 that may be responsible for both the PKD and the infantile convulsion. So we will talk about the molecular genetics about PKD. The hypothesis that the clinical overlaps came from the genetic overlap hasn't been documented until the gene PRT2 has been identified. There are two papers come out very closely that in 2011 and 2012 to prove that PRT2 is the causative gene for PKD with or without infantile convulsion. In Taiwan, our group previously documented that PRT2 is actually the most common causative gene for PKD. In familial cases, it accounts for more than 60%. And in sporadic cases, it accounts for more than 30%. Also, our patients have a hotspot mutations, and we suppose it's likely to descend from a single common ancestor. In the past few years, we keep recruiting more patients, and now the cohort should be in the largest series in Taiwan. And still the same conclusion, PRT2 still accounts for the most common causative gene in PKD. And the hotspot mutation takes more than half of the PRT2-related PKD. But what's the importance? Because PRT mutations is associated with some clinical phenotype. We know that the patient tends to have an earlier onset, and the environment tends to be bilateral. The frequency will be higher, and the attack duration will be longer. Of course, they tend to easily have a positive family history. In another series with a larger sample number, the conclusion remains the same. And also, carrying a PRT2 mutation, that means that you may have a better response to the carbamazepine. It's a sodium channel blocker we use to treat epilepsy. You can see that for the patient with PRT2, you can get almost completely remission if you take the treatment. But for the non-PRT2 mutations, the complete remission rate is lower. In our patients, in our cohort, the phenomena remain the same. For the PRT2 patients, when they start to take treatment, almost everyone can reach 0 attacks. But for the non-PRT2 PKD, the frequency can be decreased, but significant proportion of patients still have some attacks. So also, PRT2 mutation can cause other paroxysmal neurological disorder. If you know it's caused by a PRT2 mutation, you can still give him a carbamazepine, and the patient still can have an excellent treatment response. So for a heterozygous PRT2 mutation, it's easy to treatment. But when it's come to homozygous mutation, the phenotype will be much more severe. In addition to PKD, the patient will be complicated by mental retardation, episodic ataxia, or severe epilepsy syndrome. So now we call the PRT2 is related to in a wider paroxysmal disorders. PKD is the most common clinical phenotype, but also some other paroxysmal movement disorder, hemiplegic migraine, episodic ataxia, or seizure disorder. When it comes to homozygous mutation, the phenotype will be much more severe. But what's the function of PRT2? Initially, we don't know yet. We just know it's expressed on the cell membrane. It's a membrane protein. So we want to use the mutation we identified in our patient to study its function. We found that PRT2 is expressed in both the presynaptic component and the postsynaptic component. And in the mouse brain, it's colocalized with the SNAP22 in the tip of an excitatory spine. We don't know what's the function of the PRT2, but we know SNAP25 is a critical molecule involved in the excitatory transmission. So we also found that for many of the premature truncating mutation, the protein will failure to target membrane. They stay in the cytosol, so that those their function as a membrane protein. But for the point mutation, they can be expressed on the membrane, but the expressed protein is significantly reduced. Also, we accept the knockdown model. We use two different kinds of HR protein. Normally, in the neuron will migrate from the ventricular zone to the cortical plate during the developmental process. In the two knockdown models, the neuron stayed in the periventricular zone. But if you give them the wild-type PRT2, the neuron will migrate up to the cortical plate. So the neural migration is delayed but can be reversed. If the mice grows up, we can find that they have significant decreased dendritic spine density. This is a non-pathological phenomenon in many neuropsychiatric disorders, including some developmental delay, mental retardation, or autism. So based on the above findings, we propose a model of a PRT2 function. Normally, it will have the neuron to migration to the cortical plate, and it will express in the synapse to facilitate the synapse transmission. If the PRT2 loss of function, the neuron will stay or delay the migration, and the expression in the synapse will severely decrease. Finally, it will cause the synapse dysfunction. And this above work was collaborative work with Professor Junwen Cai at the National Yangming University. Then we also noted for many point missense mutations, they cluster in the C-terminus of PRT2. So we hypothesized that the C-terminus may be critical for the protein to target to the membrane. So actually, we compared with the other benign variant. The pathogenic variant caused significant decrease, particularly in the transmembrane domain. And the work, again, collaborated with two Dr. Cai, Professor Junwen Cai and Dr. Menghan Cai at Chang'an Memorial Hospital. So now we know that PRT2 is a key molecule involved in the neurotransmission machine. It interacts with many of the other molecules to facilitate the synapse transmission. So I think many people in the audience have already come up with the idea. Actually, synapse dysfunction is very important for the occurrence of an epilepsy seizure. The epilepsy seizure is due to the imbalance between excitation and inhibition. The hyperexcitation and the loss of inhibition will cause the occurrence of an epilepsy seizure. So since we now know that PKD and epilepsy have genetic and clinical overlap, and we know epilepsy is a brain networking disorder caused by the disinhibition and the hyperexcitation, is it possible PKD is also due to brain network dysfunction? So we want to look for the brain networks in PKD. We use the MEG, the magnetoencephalography, together with MRI, so that we can localize and measure the function of the neuron in different brain regions. Also, we can take advantage to detect the temporal dynamic change and to compare synchronizing the function of different regions. So the work is collaborated with Dr. Furong Xiao at the National Yangming University. First, we found that in PKD patients, they have a reduced post-movement cortical inhibition. Normally, we have a measure the event-related synchronization we call the ERS response. For the case control, we have a very good response, that very sharp the waveform. But for the PKD patients, you know the waveform is much, the peak is much reduced and the time latency is a little bit prolonged. That means the neurons, the response, lost their synchronization. And actually, the post-movement cortical inhibition is a kind of a brain function to tell our brain that stop to respond to some unnecessary stimulation. But in PKD patients, they lost the inhibition, so they have some response. This may be the clinical phenomenon we see the involuntary movements. Also, we noted that in addition to a movement disinhibition, there is abnormal somatosensory synchronization between two hemispheres in the PKD patient. And the treatment with carbamazepine can relieve the phenomenon. So we found that the abnormal sensory gating is mainly expressed by the gamma oscillation. The gamma oscillation actually is arise from the GABAergic inhibiting neurons. It's very critical in the thalamocortical circuits. They receive the sensory input and activate the lower brain center, then rely to the primary somatosensory cortex. By the MRI with MEG, we can document the sensory physiology signal is located in the primary somatosensory cortex. Actually, the role of this synchronization gamma oscillation is responsible for sensory gating. It's an important mechanism of automatic cortical inhibition. It filters some sensory input and lets our brain don't need to keep responsive to this stimulation. So again, we found that in the sensory processing part, there is this inhibition in PKD patients. So our study is to recruit 90 patients, and they are all freshly diagnosed. That means they did not take any medication, so we can get rid of the interference of pharmacotherapy. And also, we first want to look at the genotype, the correlation with the genotype and the neurophysiological change. We found that in PKD patients, compared with the case control, the peak frequency is significantly decreased, but the latency is significantly prolonged. That means they cannot respond. They start at the same time and cannot stop at the same time. They lost their synchronization. We also measured the power ratio of the second response to the first response. Normally, we will inhibit the second response, but we found that in PKD patients, the power ratio increased. That means the second response did not decrease enough. So we found that in PKD, the attenuation of synchronization and the suppression of the gamma oscillation. And we look at the difference between PRT2 PKD and the non-PRT2 PKD. We found that in PRT2 patients, the peak frequency is significantly decreased. So we think that the pathogenic PRT mutation were associated with the reduction in the peak gamma frequency. So we try to connect the molecular biology, the synapse dysfunction, we cause a neurophysiological change. That's the attenuation of somato-gamma synchronization. So they will cause the brain function to have this inhibition of sensory gating. Clinically, we will see the involuntary movement triggered by postural change. So we think the attenuation of the cortical inhibition may be a neurophysiological feature of PKD. And the interrupt sensory gating, together with the reduced post-movement inhibition, we document that PKD is a network disorder. It's not only limited to the motor, but also involves the sensory processing pathway. And also, the gamma oscillation will link to the GABAergic inhibited neurotransmission. So we proved that there is a common pathogenesis between PKD and epilepsy. This also makes the rationale why anti-epileptic drug may work with PKD. So the loss of PRT function will lead to synapse dysfunction and indirectly interrupt the GABAergic transmission. So now we know that there are some common pathogenesis to explain the genetic and clinical overlapping, and we know the brain network there is some dysfunction in PKD. So since there is a brain network dysfunction, we are eager to see is there any brain morphologic and functional change in PKD. So we use functional MRI to look for the local morphologic change, local functional change, network and the regional change. And we recruit 20 newly diagnosed PKD. Again, we want to get rid of any treatment response interferes, but we follow their MRI from the pre-treatment to post-treatment period. Among them, there are 4 patients carry a PRT2 mutation, and this work was collaborated with the Professor Yu Wu in the National Yangming University. This is our scheme of PKD MRI study. We look at the morphological, we look at the DTI analysis, and also we do the functional analysis and the network analysis. Let's first look at the morphological analysis. We measured the cortical volume and the fractional dimension. You can see in PKD patients, most of the regions, they have significant decreased cortical volume over fractional dimension. Actually, these regions are involved in the basal ganglia thalamus cortical circuit. Also very interesting, we found that inferior orbital front cortex, it has significant decreased fractional dimension, although this cortical volume did not decrease. This region is responsible for emotion, depression, and decision making. So it may partially explain that anxiety and depression has influence on the frequency and severity of PKD. Also, in contrast to other regions, we found that cerebellum, it has significant increased fractional dimension. Please keep in mind the finding. We will come back to discussion the finding later. So then we look for the fiber track DTI parameter. This is to look for the track, also for the connection between regions. And one of the important involved track is the superior longitudinal fasciculus, the SLF. Its connection to the superior parietal cortex actually is where the somatosensory cortex locates. So it's responsible for the body part of the, we call that it constructs the body mapping for us. And also connect to the supplementary motor cortex and the premotor cortex. So it's involved in the regulatory motor behavior. That means it helps us to do the test to select among the competing motor tasks. If we lose the selectivity, then we'll lose the inhibition. Our brain will be very busy in response to a lot of different response. And then we look for the local functional analysis. That means that we look for the regional coordination or homogeneity. If the coordination is good, that means the regional function is good. Again, we see in PKD patients, most of the parameters are decreased. That means that a hypofunction, particularly in cerebellum and the orbital frontal gyrus and peculius area. If you put them together, it's echoed to the somatosensory network attenuation, which we have found that in the MEG study. Then to compare the patient who has severe frequent attacks, so they need to take a drug regularly, and those who have milder symptoms, they don't need to take medication regularly. And we found that for those who need to take medication regularly, their cerebellum have significantly lower function in all the parameters compared with those who don't need to take the drug regularly. And again, we look at any difference in PRT2 carrier. You can find that the same area, very similar. Again, in the cerebellum and the temporal lobe, we found some abnormality. And actually, these regions are closely associated with a good response to oxcarbazepine. This is another anti-epileptic drug we treat for PKD. So we put the picture together for you to comparison them. We have known from clinical research study, PRT2 mutation are associated with a better treatment response to carbamazepine. Now we found in the functional MRI, their target region are very similar. So maybe it's clinically important to identify a PRT mutation in a patient with PKD because they can benefit significantly by the treatment. Also, the cerebellar hyperexcitation may play a key role in the pathogenesis of PKD. So we will talk a little bit more about the role of cerebellum for PKD. Actually, we now have the mouse model for PKD. It's the study from China. In the mouse model, they found that when the PRT2 is a mutant, some molecule in the like SNP25, the VAMP2, they are important molecule in the snare complex for physical transmission. And also the physical darkening, they have abnormal increase, but the increase is not good. For the real functional monomers, actually, the mutant PRT2 mouse have significant decrease. And then they try to do selective PRT2 deletion in different brain regions. And they found if they did a specific PRT deletion in the cerebellar ganglion cells, the phenotype is perfectly captured what we see in humans. And they did the optogenetic stimulation for the granule cell in the cerebellum and found a transient increase and then suppression of the Purkinje cell firing. When treated the mouse with the carbamazepine, the attacks will be relieved and the firing will disappear. So now we have already know that cerebellum maybe play a very important role in different kind of abnormal movement, including dystonia, chorea, and tremor. And all this can be seen in PKD patients. So cerebellum may be an important target for PKD. So in a short conclusion, from the clinical semiology, from the molecular genetics, and also from the pathophysiology, we think that synapse dysfunction is a core pathological process in PKD, and it will cause the aberrant motor and sensory network disorder. And also, cerebral hyperexcitation plays an important role in the pathogenesis in PKD. All these neurological and physiological changes can be the target for even drug treatment or neuromodulation. So this is how we approach the motor circuit of PKD. We thank for all the patients and the research collaborators, and also thanks for the funding support from NHRA and the MOST in Taiwan. Thank you for your attention.
Thank you. Is there any question from the audience? Okay. May I have the question? How do you explain the clinical cause of PKD? I mean, the patients usually have a peak frequency of attack, maybe at the senior high school or university period, and then it decreased gradually. And in my opinion, my experience, some patients suffered a normal attack after maybe 35 years old. So in your study, the dysfunctional circuit, can it explain the clinical cause?
This is a very good question. I will first use the evidence from mouse. Actually, the initial finding of PRT2, they find that if you completely knock out of PRT2, the mouse still can survive. So PRT2 is not related to survival, but it only expressed in some period during the neurodevelopmental of the mouse. And the expression will be lower when the mouse grows up. So it may partially explain that the function of PRT2 is not so essential for survival, but it may play some important role in the neurodevelopmental. And in our knockdown mouse model, not knockout, but knockdown, it will delay the neurotransmission. But finally, the neuromigration was still developed, but it just delay in time course. So maybe I think the functional PRT2 is related to certain time. It's not a function in all lifetime. It's just it just plays some role in certain time of neurodevelopment. And if the neurodevelopment is important for the motor sensory circuit development, maybe the patient will have the symptom. And also in our MEG and the functional MRI study, we all see that this abnormal phenomena can be reversed or improved by medical treatment. That means that's not a permanent structural damage. That's some functional modulation or functional influence. So by treatment, the abnormal phenomena we see in MEG or MRI, actually they can be improved by medication. And also I agree with the chair that for some patients, they still have frequent attack even after 30 years old. And also a minor part of patient, they need to take medication for a long time. I think that's partly related to genotype. We just know the most common part is PRT2 mutation. But for the non-PRT2 mutation, actually it's a heterogeneous group. Their genetic background may be quite heterogeneous. So, so in those non-PRT2 patients, in our study, for non-PRT2 mutation, the patient, non-PRT2 patients, their symptoms may be last longer and treatment response is not so good. So I think that may be due to other genetic background. Also, PRT2 is a disease determined partially by the genetics, but also very important to the interaction with the environment. That means the trigger factor. So different trigger factor or different environment will also shape the severity or frequency about the patient's symptom. Yeah.
Yes, please.
I just wondering why this kind of patient always responds to sodium channel block, not other anti-epileptic drug. If your cellular model and a model can help us.
We are still working on this because we found that the GABAergic disinhibition, but we think it's indirectly impact of the sodium channel blocker. Because for sodium channel blocker, is to inhibit the sodium channel is to reduce the hyperexcitation. And PRT2 is mainly expression in the tip of excitatory spine. So this maybe can explain the treatment response. And also to reduce the, the phenytoin. The phenytoin, maybe sometimes it can also work on the inhibitory interneuron of the sodium channel. One of the main sodium channel responsible for in the CNS is SCN1A, the 1A channel. And the 1A channel is mainly expressed in the GABAergic inhibitory interneuron. So for phenytoin or carbamazepine, they can work on the sodium SCN1, the 1A channel. Maybe that's some part of the pharmacotreatment evidence. But I believe there's still some link because there's still some steps between the GABAergic inhibition and the hyperexcitation. We still need to dig out the association. Yeah.
Okay, because the time is up, I need to close this session. And on behalf of the Taiwan Movement Disorder Society, I will present a certificate of appreciation to Dr. Liu for her contribution to this Congress.
Hello everyone, and welcome to today's talk entitled BrainSense Technology, Why BrainSense? My name is Kirk Finnis, and I'm the Global Training and Education Strategist for Medtronic DBS. So for today's presentation, we'll start by really quickly summarizing what exactly is BrainSense. And then, in the context of Parkinson's disease, we'll move on and examine why you might want to incorporate BrainSense into your practice by highlighting some of the challenges of understanding exactly what a Parkinson's patient is experiencing when they're not in the clinic. And we'll learn how BrainSense can potentially give you that missing insight into what the patient is experiencing every day, so you can be better informed when that patient is back in the clinic and in need of therapy adjustments. I'll also provide a high-level overview of the 4 key features of BrainSense and what each feature is capable of. And now the goal here is not to dive into the nuts and bolts of using BrainSense. That's actually the content of future webinars, and I hope you can join us for those. But rather, I want to give you an impression of what these features do and why they may be important to you. And not to worry though, like I said, we'll do a deep dive on each of these features in subsequent educational sessions. We'll then close out this talk with some instruction on how you configure BrainSense. And this is a quick but necessary configuration step that fine-tunes BrainSense so it's custom tailored to an individual patient. BrainSense Setup, it's called, and once complete, effectively opens the door and unlocks all of the power of BrainSense technology for that particular patient. So that being said, what exactly is BrainSense? Well, BrainSense describes the process of chronically sensing, recording, and streaming local field potentials directly from the patient's brain. And these LFP signals can contain valuable information about a patient's disease state, symptoms they're experiencing at the moment or even in the past, and may provide an objective measure of just how efficacious a current treatment paradigm really is. This is unprecedented insight into the activity of a DBS patient's brain that we've just never had before. The Percept IPG is capable of recording, processing, and storing brain signal data and patient-triggered event data directly onto the device. And this means that the patient's brain data travels with the patient. And the sensing capability can be active regardless of patient disease state or target anatomy chosen. But do keep in mind that signal levels will vary depending on the brain target, the patient disease state, and other factors like medication levels. And changes in LFP signal power can then be correlated with symptom fluctuations, ultimately providing more information for you when making changes to stimulation settings or the medication strategy. As the managing physician, you can review BrainSense data using a convenient timeline interface. It's much like a calendar that contains all of the time-stamped BrainSense data. And you can download saved LFP data directly from the device, or you can live stream LFP brain signals in real time directly to the clinician programmer when the patient's in front of you. And all of the saved and streamed data can be exported from the tablet for later offline analysis. And of course, Percept and BrainSense are fully compatible with all Medtronic DBS leads. Before we look at the specifics of each BrainSense feature, I think it's important to appreciate why you might want to utilize BrainSense to begin with. And I think we all understand that nobody wants to spend any time learning a new technology unless there's an obvious and perceived value or clinical benefit. So let's look at that. We'll start with the Why BrainSense section to establish exactly where BrainSense can fit into clinical practice and the potential value it can bring. And even though BrainSense is agnostic to disease state or indication or even target anatomy, we'll focus on Parkinson's disease patients for the moment. So I don't think it will come as a surprise to you all that managing Parkinson's disease patients can get quite complex. Their symptoms are dynamic, and when therapies are combined, like DBS along with medication, they can sometimes interact in unpredictable ways. And add to this fact that Parkinson's disease is a progressive disease. The symptoms of these patients are always evolving. And so the graph on the upper right is no doubt familiar to you all, as it shows that typical pattern of patient fluctuations that often occurs. So patients oscillate between on states where they're asymptomatic and off states where symptoms are at their worst, and on states with drug-induced dyskinesias. And I think we're also all aware that DBS therapy, of course, is used when symptoms cannot be adequately controlled by medication. When the therapy is on, you can see the fluctuation curve flatten somewhat, so it's predominantly in the on portion, which provides the patient that improvement in motor symptoms, the increased on time, and an improved quality of life. And the goal of stimulation is to keep the patient in that middle on zone, but here's the crux. We have really no way of knowing how much time a patient spends in this on zone while they're outside of the clinic, or just how effective our programming is throughout the course of an average day. We also know that side effects from stimulation and drug therapy combined can be challenging to manage, especially when they're occurring while the patient is out of the clinic, which, let's face it, is about 99% of the time. So there's a lot going on with a patient's therapy state while they're not visible to you, the managing clinician. When managing a fluctuating patient, many physicians will rely on the existing metrics of evaluating the patient's symptoms, and these include the UPDRS and patient motor diaries, which both could be considered fairly subjective. Also, motor diaries are a burden to the patient, and as a result, they typically have low compliance and often don't contain enough data points to really paint a complete picture of the patient's symptoms outside of the clinic. And motor diaries, being the de facto standard, absolutely have some significant limitations. And when you think about it, missed medications or random patient-initiated changes in stimulation and missing diary entries, you're left with an incomplete and often inaccurate overview of symptoms a patient experiences when outside of the clinic. And clinical decisions regarding medication or stimulation adjustments must be made on in-clinic observations, given the suspect accuracy of these diaries, and also on what the patient is capable of reporting to you directly. So you can see the red line in the graph is what you may think or infer from an incomplete motor diary versus what may have actually occurred. And this is where Percept PC and BrainSense comes in to help fill that information void. So we're providing the data that managing physicians have historically never had, and that's a record of potentially what's going on with the patient outside of the clinic. And so Percept can collect and record chronic brain signals, or LFP signals, from DBS implanted anatomy while the patient is in or out of the clinic. And we see here a representation of two DBS contacts on a 3389 lead that are recording LFP data from the left STN. And greater than 15 years worth of research strongly suggests that LFPs are not just academic, they can be a known correlate of patient symptoms. And these LFP brain signals, and we'll focus on beta here for Parkinson's disease, have been shown to correlate with patient disease state, including symptoms, symptom fluctuations, and efficacy. And efficacy not just of the DBS, but also for the patient's medication. The graph on the right shows what can be best described as a linear relationship between beta LFP power and a PD patient's motor symptoms as measured by UPDRS. And so to the right of the graph, the patient's motor symptoms are significant and the corresponding beta power measurement is elevated. And to the left side of the graph, the patient's motor symptoms return to the on state and the beta correspondingly decreases. The brain signals recorded using BrainSense can contain valuable information about that patient's symptom fluctuations and their overall disease state while outside of the clinic. And understanding the fluctuations in beta for a Parkinson's disease patient may provide that missing information and prove invaluable in managing adjustments of both stimulation parameters and drug therapy. Now you may have noticed in the two graphs on the right that the yellow line at the bottom, which represents beta LFP power fluctuations over time, is effectively a mirror image of that standard symptom graph I showed earlier. And this is because of that linear relationship. When beta LFP power increases, the patient is pushed into an off state. And conversely, as beta LFP power decreases, the patient is freer to move and is in an on motor state. So if a physician with access to BrainSense information can first document these changes in LFP power and then program the stimulator to minimize them, like the beta fluctuation we see at the bottom of the screen, and flatten that curve out, that physician can potentially maximize stimulation efficacy using feedback from the patient's own brain. And so here's an all-encompassing summary of BrainSense, and I'm just going to read this verbatim. BrainSense provides an objective, data-driven method for monitoring brain signals that may define certain symptoms of a disease state to better inform the clinician when making programming and medication changes. Now notice I didn't mention beta once in that message, and beta is definitely important. Beta is a robust biomarker for achinesias and rigidity symptoms for many akinetic rigid Parkinson's disease patients, but these patients are often not the only DBS patients in the population of patients that you manage. There's a lot more opportunity out there than just this one patient subgroup. And our European customers, which we launched in Europe back in January, have been using Percept and BrainSense in implanting in epilepsy patients, in tremor-dominant PD patients, essential tremor patients, OCD patients, and dystonia patients, among others, and using BrainSense to investigate different frequencies of interest for each. So please keep in mind that Percept and BrainSense are not just for your akinetic rigid PD patients. It just so happens that that is the demographic that we currently understand the best. And I'd encourage you to reach out to our medical affairs team should you have any questions on LFPs and physiologic biomarkers for other conditions. And what you may also find interesting is that we now have a DBS system in Percept that can potentially provide valuable insight into not only how well the DBS is working on patient symptoms, but also the efficacy of the medication a patient has taken, simply by looking at what happens to that elevated beta power over time after a patient takes their medication. So to summarize the answers to that question, why BrainSense, I think we can say that BrainSense provides insight into your patient's brain while they're not in the clinic, which is, again, roughly 99% of the time, and may generate usable data to better inform your treatment strategy. And BrainSense enables the automatic creation of an electronic patient diary that's stored directly on the device. And this includes patient-triggered events so that they can exactly mark when something unusual happened, or when they experienced a specific symptom, or when they simply took their medication. And in addition to this, BrainSense will record and store LFP signals over time directly onto the device and without patient involvement. So trends in the LFP data over time may then be assessed and correlated with changes in patient symptoms and possibly even disease progression. And like I mentioned, we talk a lot about beta given the sheer volume of supporting evidence available, but remember that BrainSense permits monitoring of any LFP frequency band between 0 and 100 Hertz for up to 60 days. So this could obviously include delta or theta, alpha or gamma bands, depending on the interests of you, the clinician, and the patient's disease. BrainSense can also provide a summary screen that analyzes much of the recorded data for you based on user-defined thresholds, so you can see at a glance just how often a patient was potentially well-treated or undertreated while at home, depending on how elevated their beta signal was. And we can delve into the details in this in a future session. So now that we've discussed why BrainSense and the why BrainSense question, let's look at BrainSense technology and the 4 primary physician tools that it contains. So the first feature we'll look at today is BrainSense Timeline. And BrainSense Timeline is much like your home base on the tablet for all of your recorded sensing data. So this is where you'll find all of the data that's been collected while the patient was out of the clinic, where it's conveniently laid out in a calendar-based format. Now, we're not going to go into great details on its usage today, as that's a topic for another webinar, but let's quickly enlarge this screen grab so that we can appreciate some of the details that are available to you. So in this screenshot, we can see the resulting beta LFP brain signal in the topmost graph that was recorded on a specific day, and it's October 14th in this example, while the patient was outside of the clinic. And hopefully you recall that wavy yellow LFP line we discussed earlier that may correlate with the patient's symptoms, and this is really what we're trying to find in Timeline. And we're seeing here how beta fluctuates over one complete day from midnight to midnight. You'll notice that time is on the x-axis and the power of the beta signal is on the y-axis. And each one of those little yellow dashes is a 10-minute average of the LFP beta power collected throughout the course of the day. And based on what we know of the linear relationship of beta power and symptoms of akinesia and rigidity, we can begin to assess when the patient was most likely symptomatic at the peaks of the graph or asymptomatic at the valleys. And BrainSense Timeline allows us to save up to 60 days of recorded LFP data and look for trends that appear over time. So using this calendar view format, we can easily change from one day to the next, either with the swipe of a finger or by clicking directly on the date. So if a patient reports that their symptoms were particularly bad, for example, last Thursday, you can simply click on that date to see what was recorded. And we can also see the patient's active stimulation setting in the bottom graph. And it will tell us if they've increased or decreased their stimulation within their visible limit lines, that pale blue lines above and below the orange line, that shows the DBS stimulation amplitude level. So with time